Good morning, everyone. My name is Brittany Green, Communications Director of the KAT6A Foundation. Welcome to our very first KAT6A clinic. We are delighted to have you all here. We know many of you have traveled long distances to be here to support KAT6A, and for that we are very grateful. I wanted to take a moment to thank Dr. Harris for organizing this clinic and making this possible, Dr. Bjorsen for speaking to us about frontiers and therapies for epigenetic disorders, and also Dr. Kelly for traveling from Massachusetts to share the data he has been collecting and analyzing on our children. Also a big thanks to the KAT6A Foundation team for pulling all of this together and putting everything into place. We have today from the Foundation board members and committees, Jordan Muller, Chairman, Emal Najm, President, Amy Redson, Website, Natasha Esper, Scientific and Research, and Miranda Lentine, Social Media. We all know how rare and extraordinary our kids are, and with the help of these amazing doctors, we are hopeful that soon we will have a lot more information about the KAT6A syndrome. We are looking forward to meeting and getting to know all of you. It is truly a dream come true to have so many of our KAT6A family together here today. Yay.
Also, Rick, we have Karen, board member. Karen Ginsburg.
Okay. Oh, I'm sorry.
Hi, everybody. Thank you so much for everybody who came from near and far to do this. We really appreciate everybody being here. We're very excited about this here at Johns Hopkins and Kennedy Krieger. KAT6A, as you all know, is a relatively newly discovered syndrome, and we know that you all have been sort of patients in search of a medical home, and so we are really hoping that this can be the start of you all meeting each other, and you all meeting some of us, and us taking a really good team approach to helping these very special and wonderful children as they are children and as they go on to be adults. I am Dr. Harris. I am a neurologist and neurodevelopmental specialist, and I work here at Kennedy Krieger and also in the epigenetics clinic at Johns Hopkins. So I am very interested in KAT6A. It has a lot of overlap with lots of other things that I have interest in, and am very excited that you all came to this. Here also we have Dr. Bjorsen, who many of you all have talked to before. So he is here in the epigenetics clinic with us at Hopkins. Dr. Bjorsen has dual appointments here in Baltimore and also in Iceland. So he is around and here and a part of our clinic, but not always present physically in Baltimore. And then we also have Dr. Jill Farner. She is also part of our epigenetics clinic. She is here at Hopkins in the clinic, and also has a strong interest in KAT6A and disorders that are related to it, and I was very excited to see you all. And we really appreciate Dr. Kelly coming all the way down. Dr. Kelly used to be around with us here all the time, but has moved north where it's colder. And we are glad he could come back to be with us today and share with us his work and everything that he's doing with you all and for you all also. So a quick word about kind of how the day is going to go. So we have 3 relatively brief talks. I'm going to talk a little bit about just KAT6A, the disorder, what is known about the patients clinically, and what's not known, and what we need to continue to discover. Dr. Kelly is going to talk about his research and data he's collected in a large number of patients he's seen or collected samples from with KAT6A. And Dr. Bjornsson is going to talk about some frontiers in epigenetic therapy, which KAT6A is an epigenetic disorder, and sort of where we think and hope that we are going in the future. And then after that, we're going to have a lunch, and lunch is informal, as is the rest of the afternoon. And what we really want with that time is to make sure that you all get a chance to talk to each other, because that's some of the most important things about gatherings like this, is you all learning from each other and gathering support from each other, and kids of different ages sort of getting to meet each other as well. But also we will be around so that you all have an opportunity to sort of informally talk to us and mingle with us, and we get to meet you. So hopefully that works out, and then yeah, it should be a great day. We'll wrap up around 2:30 because they're kicking us out of here at 3. And you know, hopefully, if this is all successful and you all enjoy it, we can do another one of these in the future. So with that, we'll get started with the talks. So you heard me say, and I'm sure you've heard before, about KAT6A being an epigenetic disorder. So what exactly does that mean? So epigenetics is basically all of the stuff that happens to the DNA after it's already there. So the DNA code is written, it's down, it exists, but then things that happen to it after that enable it to be read or turned into proteins as it needs to be. And that's what epigenetics is. So KAT6A is a special type of epigenetic syndrome, which is a syndrome that creates a modification to histones. Histones are proteins that inside the cell, the DNA gets wrapped around. It gets wrapped around very, very tightly in the nucleus in the center of the cell, because the amount of DNA that we have in every single cell is incredibly long. And so these proteins help it to be compact and be able to exist in every single cell. However, they are not just, you know, rocks that these get wrapped around. They are actually active proteins that have a role in opening and closing to provide this DNA the ability to perform the functions that it's there to function. And so the KAT6A protein actually has a role in modifying these histones. So what it does is it places a mark on these histones that allows the DNA to open appropriately to be read and transcribed. So thinking about this a little bit further, we can group these epigenetic modifiers into 3 main categories: writers, erasers, and readers. So writers are things that add marks to these histones, and these marks can say open up, or they can say close down. There are proteins that erase the marks, so take them away. And then there are other proteins that read the mark. So once the mark is put on or taken off, it looks and says, what is this mark telling me to do? So KAT6A is actually a KAT stands for K is a symbol for lysine, which is an amino acid. The AT is for acetyltransferase, and then the 6A just lets us know which one it is. And so what that means is that this protein actually moves an acetyl mark onto these histones and tells the histones what to do, how to be open or closed appropriately. So you can understand why, if something goes wrong with that particular protein, then a lot of things can go wrong later in the body. Because your DNA is present in all of these cells, all of your cells, and if it's not there appropriately to be written or used the way it's supposed to, then you have many different problems in many different parts of the body. We know that this particular protein, KAT6A, is especially prevalent in the brain, and so that is why that is the focus of a lot of the clinical features that we see in this syndrome. So just to give you an example, you can see that KAT6A is there on top, and there are a number of other proteins that work with it on these histones. They work together in complexes.